So we have started discussion of aerobic respiration. And for aerobic respiration, what we have said, just in a glimpse, I would like to revise that this mechanism of anaerobic respiration, it is completed in 4 stages. Obviously, stage 1, it was glycolysis that already we have discussed in detail. The stage 2, it will be oxidative decarboxylation. The third one, it is the Krebs cycle, or that is also called as tricarboxylic acid cycle. And the last stage, it is going to be the terminal oxidation and electron transport system. Now, actually, we have discussed the glycolysis. It is the process by which respiratory substrate glucose, it is broken down into 2 molecules of pyruvic acid along with the production of 2 ATPs and 2 NADPH2. That entire biochemical pathway we have studied. And then we have gone for the balance sheet or net gain of glycolysis, that is nothing but the 2 molecules of pyruvic acid and then 2 molecules of ATP along with 2 molecules of NADPH2. Then actually today we are going to start with the discussion of second stage, and that second stage obviously it is said to be the oxidative decarboxylation. It is called as oxidative decarboxylation. Now, to understand this oxidative decarboxylation, I would like to take you to the whiteboard so that you can understand. See, this is the mitochondria where all the remaining phases will be completed, right? And surrounding this, it will be the cytoplasm. And within this cytoplasm, we have said that the process of glycolysis has been completed. And during glycolysis, we know it is the 2 molecules of pyruvic acid, they are produced, right? Now, these 2 molecules of pyruvic acid, they have got how many carbons? 3 carbons. And these 3 carbon pyruvic acid molecule, they cannot go into the what we call it as a mitochondria. So these 3 carbon compound, they cannot be taken into the mitochondria because the channels of mitochondria, they are so small, they does not allow the entry of these 3 carbon compound. So when 3 carbon compound cannot be taken into the mitochondria, what is done? That 3 carbon pyruvic acid, it is chopped, it is cleaved, it is made small, and then it will be taken into the mitochondria. So you are getting it, 3 carbon pyruvic acid, it cannot go into the mitochondria because mitochondrial channels are small. So to allow the entry of that pyruvic acid or organic molecule into the mitochondria, pyruvic acid 3 carbon compound, it is chopped, it is broken down, it is cleaved, and then it will be taken into the mitochondria, and that process is called as oxidative decarboxylation. That process is called as oxidative decarboxylation. So let's understand this oxidative decarboxylation as a whole. What is that? Obviously, it is the oxidative decarboxylation of pyruvic acid, or simply it is called as acetylation process, because pyruvic acid 3 carbon compound is converted to 2 carbon acetyl moiety, acetyl group, and that's why it is called as acetylation process. It is the first step in the respiration during which CO2 is removed, or the first decarboxylation will occur. If you remember, during glycolysis, CO2 wasn't removed. Simply glucose was broken down into 2 molecules of pyruvic acid, but there wasn't any release of CO2. First CO2 release, first decarboxylation will occur during this process. In this step, pyruvic acid, which is generated in the cytoplasm by glycolysis, it will enter into mitochondria, but as a 3 carbon compound, no, it is not possible. So it will be converted to acetyl moiety, and then it will be taken into the mitochondria. And for that purpose, in presence of pyruvate dehydrogenase enzyme complex, pyruvate dehydrogenase enzyme complex which dehydrogenase, means it will dehydrolyze, or we can say simply it will dehydrogenate the pyruvic acid. And for taking up the hydrogen, it will be NAD which is available. By taking the hydrogen from pyruvic acid, NAD will be converted to NADH2. And then as a result of that, a 2-carbon compound will be produced that is called as acetyl moiety. Now this acetyl moiety will be taken up by a carrier molecule which will transfer that acetyl into mitochondria. That is, we can say formation of acetyl-CoA along with the formation of NADH2, and first CO2 molecule will be released. Now, actually, this formation of acetyl-CoA, it will start in the cytoplasm and it will be completed into the mitochondria. Let's see how that happens. Here you can see, once again, you are going for the whiteboard. Yeah, here it is the mitochondrial membrane. Then here it is the 3-carbon pyruvic acid is there. Now this 3-carbon pyruvic acid, it cannot enter into mitochondria क्योंकि mitochondria का channel छोटा है, वहां से 3 carbon molecule अंदर नहीं जा सकता. So this 3-carbon pyruvic acid, it undergo decarboxylation. 1 CO2 will be there and there is a formation of 2-carbon compound in the cytoplasm, and that is nothing but the acetyl moiety. Now, on the outer surface of mitochondria, there is a coenzyme, and that coenzyme is coenzyme A. Okay, so in the cytoplasm, this 2-carbon acetyl moiety, it will combine with coenzyme A, and there is a formation of acetyl-CoA. Now, this acetyl-CoA will contain only 2 carbon, and that 2 carbon can be very easily taken into the mitochondria from both the outer and inner membrane. So for transferring this 2-carbon acetyl moiety, coenzyme A is required. And then you can see it is the cytoplasm and it is the mitochondria. So both of the type of reactions of cytoplasm and mitochondria, they are connected. That's why this acetylation, it is also called as link reaction. Are you getting this? Why this acetylation or decarboxylation is required? Because 3-carbon pyruvic acid, it cannot go into mitochondria because the channels of mitochondria are small. 3-carbon compound cannot go in. So 1 carbon is removed, 2-carbon acetyl group, and for carrying that, a carrier molecule, coenzyme molecule, CoA will be there. So there is a formation of acetyl-CoA, and then that acetyl-CoA can go into the mitochondria. That is the meaning, that is the requirement, that is the basic process which is during the acetylation of CoA, right? Then actually, this process of acetylation, it is nothing but the conversion of pyruvic acid into acetyl moiety combined with coenzyme A to form acetyl-CoA. That process is called as acetylation, or it is called as link reaction, because it is linking cytoplasmic pyruvic acid into the acetyl moiety, which will be taken inside the mitochondria, right? Then if we move further and see, this acetyl-CoA which enters into the mitochondria, now it will go into the Krebs cycle for further degradation. The third step we know, it will be Krebs cycle. So acetyl moiety will be the raw material which is available for going into the Krebs cycle. Now, actually this molecule which is produced on the outer surface of the mitochondria, we call it as an acetyl-CoA. Now it is also called as connecting link between glycolysis of cytoplasm and Krebs cycle of the mitochondria, right? Glycolysis, it is taking place within the cytoplasm. Then the pyruvic acid will be produced. It will be trimmed to 2-carbon acetyl moiety. 2-carbon acetyl moiety will go into the mitochondrial matrix, where the next step will be there. It is nothing but the Krebs cycle. So this acetyl-CoA, it is an substrate entrant for Krebs cycle. Krebs cycle will start from acetyl-CoA, not from pyruvic acid. Pyruvic acid was in the cytoplasm. Usko tod diya, uska chota bana diya, acetyl-CoA bana diya. Then that acetyl-CoA will enter into the Krebs cycle. Now, during that oxidative decarboxylation of pyruvic acid, CO2 was removed and oxidation will result into formation of removal of hydrogen. Now this hydrogen, apne ko pata hai, it is always the NAD which will accept the hydrogen and it will become reduced form of NADH2, and that NADH2 will be available for electron transport system to produce 3 ATP. Baad mein. We will see that later on. Now here the two molecules which are produced during this oxidative decarboxylation, we know 1 NADH2 is equal to 3 ATP. तो यहाँ पे 2 बनेंगे तो अपने को मिलेंगे 6 ATP. So during oxidative decarboxylation, we will have 2 NADH2 that are equivalent to 6 ATP molecule in all. And then if we see the overall reaction which is taking place, the pyruvic acid plus coenzyme NAD, coenzyme CoA, and for hydrolysis water will be required, and for dehydrogenation reaction enzyme is required, pyruvate dehydrogenase complex. Now this pyruvate dehydrogenase complex will remove one CO2, it will be pyruvic acid converted to acetyl group. Then hydrogens will be removed, they will be taken by NAD, that will be converted to NADH2. And then that acetyl moiety will combine with coenzyme A to form acetyl-CoA, NADH2, and CO2 will be released. Now this acetyl-CoA, it will enter into the mitochondrial matrix for the stage 3, and that stage 3 is nothing but the Krebs cycle. But we will go on Krebs cycle later on. Let's understand this. Oxidative decarboxylation of pyruvic acid. The first question, why oxidative decarboxylation of pyruvic acid is required? The answer is the pyruvic acid will be produced during glycolysis in the cytoplasm. The pyruvic acid will contain 3 carbon, and this 3 carbon pyruvic acid, it cannot go into the mitochondria because mitochondrial membrane will have the channel that will not allow the entry of 3 carbon compound. So what is done? 1 carbon is removed from the pyruvic acid. It is converted to acetyl moiety. Now this acetyl moiety will be produced on the cytoplasmic part. In the mitochondrial membrane, coenzyme A will be there. It will take up that acetyl moiety and it will conduct that into the mitochondrial matrix, and there it will release that acetyl moiety for Krebs cycle. And only for that purpose this decarboxylation is required. Number 1. Number 2, dehydrogenation of pyruvic acid will remove the hydrogen, and these hydrogen will be taken up by NAD and it will be converted to NADH2 reduced form, which will be available for electron transport system, where we will generate 6 ATPs from 2 NADPH2. So that is the story of oxidative decarboxylation of the pyruvic acid, or acetylation process of what we call it as an pyruvic acid acetylation. So that is the first thing that we have discussed today. Have you got any problem? Yes?
No, sir.
No, sir. No, sir.
Okay. And then we will proceed on to the third part. Now you remember the first part, it was glycolysis. Number 2, it will be the wait. Yes, it is the third step. The step 1, glycolysis. Step 2, it is acetylation. And then the third step, it will be the Krebs cycle. More correctly, it is called as TCA cycle, tricarboxylic acid cycle, or even it is called as citric acid cycle. Now, very importantly, after oxidative decarboxylation, the acetyl moiety which was produced, now it will it will be transported into the mitochondrial matrix. So Krebs cycle, it is going to occur in the matrix of mitochondria, and for this process, we know the molecular oxygen, it will be available as terminal oxidant. The last electron acceptor, it will be the molecular oxygen. Now, actually, it is representing the step 2. It is representing the step 2 of aerobic respiration. In old concept, glycolysis was the part 1 and the Krebs cycle was part 2. But nowadays, in modern sciences, we consider that we can say oxidative decarboxylation of pyruvic acid as step 2. But originally, the Krebs cycle was step 2. Now, in this cycle, the pyruvic acid, the pyruvate, it is further degraded to CO2 and H2O, releasing the energy. So complete oxidation of pyruvic acid or glucose will take place, releasing the 6 molecules of CO2 and 6 molecules of H2O. Actually, this cycle, it was discovered by a great scientist, Sir Hans Krebs. Sir Hans Krebs was the first person to study the fate of pyruvic acid after glycolysis, and he studied that in the pigeon muscles. Pigeon, you know, dove. In the dove muscle, in the pigeon muscle, for the first time, he invented the metabolic pathway for degradation of pyruvic acid, and for this great work, he was awarded with a Nobel Prize in 1953, the same year when the discovery of structural model of DNA by Watson and Crick has been done. You can imagine at a time, biochemistry and plant physiology research was going on. So Sir Hans Krebs, 1937, discovered this cycle in pigeon muscle and got awarded with the Nobel Prize in 1953. Now, during this Krebs cycle, what is coming in? It is acetyl moiety, 2 carbon. Now, this 2 carbon acetyl moiety will be accepted by the 4 carbon compound, and that 4 carbon compound will be oxaloacetic acid. In the mitochondrial matrix, 4 carbon oxaloacetic acid, 2 carbon acetyl moiety, they will combine together to form a 6 carbon stable compound. Now, that 6 carbon stable compound, it is called as citric acid. So during Krebs cycle, the first stable compound which is produced, it is containing 6 carbon citric acid. That's why it is also called as citric acid cycle. Why it is called as citric acid cycle? The first stable compound is citric acid. Similarly, the various acids which are produced during Krebs cycle, so many acids we will see, but all of them, they are one thing in common. All of them will contain 3 COOH group, that is carboxylic acid group. जितने भी एसिड अपन देखने वाले क्रेब्स साइकिल में, सब में minimum 3 COOH group, 3 carboxylic acid group will be there, and that's why it is also called as tricarboxylic acid cycle. Sorry, it is a TCA cycle. Now, actually, this entire metabolic pathway of Krebs cycle, it will be controlled by some specific enzyme. It will be catalyzed by some specific enzyme. What are these enzymes? What are that pathway? Definitely we will see. But once again, we will go for the introduction of that Krebs cycle. Number 1, in the glycolysis, 2 molecules of pyruvic acid were produced. They were containing 3 carbon. तो वो 3 कार्बन कंपाउंड को माइटोकॉन्ड्रियल मैट्रिक्स में लाने के लिए उसको ट्रिम किया. It was converted to acetyl group. That acetyl group was taken by coenzyme A. Coenzyme A, acetyl group, collectively they are forming acetyl CoA, and then that acetyl CoA will be brought into the mitochondrial matrix. Mitochondrial matrix में आने के बाद में coenzyme अलग हो गया, acetyl group अलग हो गया. ये acetyl group का क्या करने का? That will be Krebs cycle. So it occurs within the matrix of mitochondria. In presence of molecular oxygen, molecular oxygen will be acting as a terminal oxidant, last electron acceptor. In olden days, it was considered as a second step of aerobic respiration, but now we know step 1 glycolysis, step 2 acetylation, step 3 will be the Krebs cycle. It is also called as citric acid cycle, or even it is called as TCA, even it is called as Krebs cycle. Why Krebs? Because Sir Hans Krebs, he discovered it in the pigeon muscles, and that's why it is called as Krebs cycle. He has got Nobel Prize for that. Then the first stable compound In the Krebs cycle, it is a 6 carbon citric acid. तो पहला स्टेबल कंपाउंड साइट्रिक एसिड बन गया, तो नाम दे दिया साइट्रिक एसिड साइकिल. सिमिलरली जितने भी सारी ऑर्गेनिक कंपाउंड यहाँ पे अपने को मिलने वाले हैं, ऑल ऑफ देम विल कंटेन एटलीस्ट 3 COOH ग्रुप, बोले तो कार्बोक्सिलिक एसिड ग्रुप. तो नाम बन गया ट्राई कार्बोक्सिलिक एसिड साइकिल, और सिंपली इट इज कॉल्ड एज TCA cycle. And each and every step of this biochemical pathway, it will be catalyzed, it will be controlled by some specific enzymes, right? Once we do that, then we have to go on to the actual steps that are involved in the Krebs cycle. And this is the representation of the Krebs cycle as a whole. ना यहाँ पे तुमको पूरा का पूरा Krebs cycle along with the decarboxylation, oxidative decarboxylation will be shown. See what I am going to show you. Number 1, यहाँ पे देखो, this is the respiratory substrate glucose containing 6 carbon. The step 1, it is the glycolysis, and this glycolysis has resulted into formation of 2 molecules of pyruvic acid containing 3 carbon. Now this is the step 1 that is called as glycolysis. Now this pyruvic acid which is produced at the end of glycolysis, what happens to it? It will go for the second step. And in second step, it is, we know that the second step is oxidative decarboxylation, because this pyruvic acid, it undergo decarboxylation, बोले तो removal of 1 CO2, और 1 CO2 निकल गया, 3 में से तो 2 बचे. That 2 carbon compound, it is called as acetyl moiety. That acetyl moiety will combine with coenzyme A, and there is a formation of acetyl CoA. Now this acetyl CoA will enter into the mitochondria for Krebs cycle, and during that, removal of hydrogen from pyruvic acid will give rise to NADH2. So these are the 2 steps we have represented there. या दोन्ही स्टेप आपण इथे रिप्रेझेंट केलेला आहे. आणि मग हा acetyl moiety which comes into the mitochondrial matrix will undergo the Krebs cycle. So that is the basic idea. Step 1 यहाँ पे दिखाया है, glucose undergo glycolysis to form 2 molecules of pyruvic acid. Step 2 oxidative decarboxylation, ते पण इकडे दाखवलंय आपण. Pyruvic acid undergo oxidative, hydrogen removed, taken up by NAD, NADH2 is formed. Decarboxylation, removal of CO2. It will result into formation of 2 carbon compound acetyl moiety. That acetyl moiety will combine with coenzyme A, and there is a formation of acetyl CoA. Acetyl CoA एक बार mitochondrial matrix में आ गया तो उसके साथ में बनेगा Krebs cycle, and that Krebs cycle, it will be a long metabolic pathway that we will see later on. पण त्याच्या आधी पहिल्या 2 स्टेप कळल्या आहेत का? ग्लाइकोलाइसिस एंड ऑक्सीडेटिव डिकार्बोक्सिलेशन. मुझे ये बताओ, अगर यहां तक समझ में आ रहा है तो we will go further. If not, we will go for some doubt clearing session.